Welcome back students. We are going to talk about various other examples of the multiple alleles. And today now the example that which we are discussing in this session is the ABO blood grouping. In the case of humans, we know Landsteiner, he discovered the ABO blood grouping in the case of humans, and we have different types of blood groups, group A, group B, group AB, as well as group O. And the classification of these groups, blood groups, are mainly due to the presence of certain markers on the RBC. In the case of group O, we see that we have certain antigens called H antigens on the surface, but we do lack the presence of certain sugars on the surface of these H antigens. And the RBCs which have the H antigen but do not have the sugars present on them are referred to as the group O antigens. Let it be all the blood groups A, B or AB, they all have commonly the H antigen which has been depicted as a line on the RBC cell. Now, if you are having a group A type of blood group, you have one type of sugar being present, A antigen, which is being present on the H antigen. Okay, and if you are having a B antigen, you have a B blood group, you are having a B antigen on the RBC cell. And if you are having a group AB, you have both A antigen as well as the B antigen on the present on the RBC cell. And we all know that if you are having a A blood group, in your plasma, you have the antibodies against B. That is, you have the anti-B antibodies in your plasma. And if you are having a B blood group, you have an anti-A antibody in your plasma. And when you're having a group AB, you do not have any antibodies against the A antigen or the B antigen in your plasma, but you have the A and the B antigen on the surface of your RBC. And when you have the group O or the blood O blood group, you have anti-A as well as anti-B antibodies in your plasma, but you do not have the A antigen as well as the B antigen on the surface of the RBC. Now, let me just show you this. What is the difference between these blood groups? As I mentioned earlier, on the surface of the RBC, there is a H antigen or the stick-like shape which is being present here. That is referred to as a H antigen. And on this H antigen, if there is a sugar which is being made up of N-acetyl galactosamine, it constitutes the A antigen. And if the H antigen has a terminal galactose residue or the sugar which is being present on this H antigen, if it is galactose at the end, it will constitute the B group. And if the H antigen of the RBC, which is being present in all the blood group, has N-acetyl galactosamine coding for the A blood group, as well as galactose which is coding for the B blood group, then the blood group will be considered to be as a AB antigen. So just look into one more thing. All the blood groups have the H antigen, which is common, which is made up of a polysaccharide. And on the tip of this H antigen, if you are having a sugar, a terminal sugar containing N-acetyl group galactosamine, that is a pink one here, that will constitute the A antigen. And if you are having on the terminal of the H antigen a galactose residue, then it will constitute the B antigen. So this is the basis of difference of different blood groups. When you are having on the RBC, on the H antigens, both A antigens as well as the B antigens are being present on the blood group, you constitute to form the AB blood group. So the difference between group A and group B is only the difference on the terminal sugar which is being added onto the H antigen of these RBCs. If N-acetyl galactosamine has been added to the terminal as a terminal sugar on the surface of the RBC, then it is referred to as the group A. And if galactose is being added, it is referred to as the group B or the B blood group. I guess now you are quite clear about what are the different type of blood groups. Now, let me ask you, how do you inherit the blood group? Or what are the genes responsible for this type of blood group variations? As it is quite clear here, if you're having different blood groups, you would have different types of alleles which are being coding for this particular blood group. Now, in the case of ABO blood grouping, it has been found the gene which has been coding for the ABO blood grouping, it has been found to be autosomal. That is, it is located on the not on any sex chromosome, but on the somatic chromosomes, and it is mainly located on the chromosome 9. And the A and the B blood groups, they are being found to be dominant over the O blood group, and at the same time A and B blood group genes are being found to be codominant. And we know that everyone has a maternal chromosome as well as a paternal chromosome, and it is based upon the presence of which allele is being contributed. Based upon that, you are able to find out the what particular blood group is being formed. In the case of A blood group, you can find that it could have a genotype of IAIA, and also a genotype of IA and small letter I. And in the case of those who are having a B blood group, they can have a IBIB or a IB small I type of genotype. And in the case of people having the AB blood group, you can see that IA and IB should be present. That is, they are being found to be co-dominant in each other. That is, when you have a maternal or a paternal gene coding for IA and another one which is coding for a IB, you will get both the genes are being expressed, and as a result you will get a AB blood group. Now, if you're having a O blood group, you can see that you have a recessive gene, small I, and that is a one which is mainly responsible for the presence of O blood group. You know what is O blood group? That is, you will not have the A antigen as well as the B antigen, and you will have the A antibodies as well as the B antibodies which are being present in your plasma. So that's about the genetics of the blood grouping. Now, in addition to the ABO blood grouping, we can also find that in the case of blood groups, we also have another factor called the RH factor, which determines whether we have a RH positive, AB positive blood group or a AB negative blood group. The presence of RHD or the RH factor is a one which is responsible for the positive or the negative nature of the blood group. Now, this RH gene is located on the short arm of chromosome 1, and it was discovered in 1940 after the work on rhesus monkey, and that's what the term RH was being denoted. Now, if you're going to look into the genes of this, you can see that in the case of RH positive, if you're having a RH positive cell, you will have the RH antigen or the RHD antigen which is being present on the RBC. And if you are having a RH negative, you do not have the absence of this product of the RHD gene on the surface of the RBC is the one which refers to as the RH negative nature. Now, these are proteins which carry the RH antigens are the transmembrane proteins, and these structures suggest that they are the ion channels. They serve as mainly as ion channels. And the RH antigen is being coded by capital letter D, C, E as well as small letter C and small letter E genes. And that is the different forms of that particular gene is being present over here, and the positive nature is always being the presence of this RHD gene is always been present in the case of RH positive people. Now, along with the RHD gene, Okay, you can have either capital letter C, capital letters E, or small letter C or capital letter E. Like that, different variants of C and E will be combined with the capital letter D gene, and it is a one which it is responsible for the Rh positive or the Rh positive nature. So the Rh factor is mainly being coded by these three genes. Okay, C, D and E. And if you are Rh positive, it means that the D gene is being present. There is no small lowercase case d, but it is the absence of the D antigen which is indicated by the small letter D. Now, if you go to look into the blood grouping, you can find that if you're having a ABO and RHD combination, okay, if you're having a mother which is having an A blood group and she is Rh positive, and if there is a father which is B positive and he's Rh positive, so you consider the genotype is capital letter D and capital letter D, small letter d, and what are the possible ways that which we can have? You can either have the offspring can have either A positive, AO, that is one positive thing, or the nature of the thing, or what is it, the Rh antigen, it can be capital letter D and small letter d from here, or it can be a B antigen from the father and the O antigen the same here. So it can be a B blood group or it can be a capital letter small letter d here. Then it can be a what? O blood group which is being present here, or it can be a Rh negative nature. In addition to this, we can also see that various other blood groups have been possible based upon the what? The genotype of the father and the mother. So you can just explore that using this particular example. And now let's move on to erythroblastosis fetalis, which is a hemolytic disease, as you all know, which is to be of much importance when a Rh negative mother gives birth to a Rh positive baby. In the first case, the baby has no problem, but when the blood of the baby happens to mix with the Rh negative mother, she produces anti-Rh antibodies after her first pregnancy. And so if she conceives the next time and if it's a Rh positive fetus which has been formed, the fetus will be damaged by the anti-Rh antibodies which have been produced by the mother. So to prevent this situation of erythroblastosis fetalis, we need to take injections or we need to take the precautionary injection so that the anti-Rh antibodies will be prevented. In addition to this, we also have a secretory type of blood grouping system. I'm just mentioning it here so that it would be coming to be as forensic students, you should also know it, because when you go to consider your body fluids like plasma, saliva, synovial fluid, nasal secretions also, there are some people who will express the antigens present on their RBC in these fluids. That is, you know what is it? Not the RBCs I'm saying. In these plasma or not sorry, in this body fluids, you can find that the antigens which have been present on the blood groups could also be present in their body fluids. And those who are having them in the body fluids, those people who express these ABO antigens in their body fluids are considered to be secretory positive, and those who do not express these soluble antigens in their secretions are considered to be as non-secretors. And so the secretions is mainly being controlled by the presence of H, E and SE genes. And what happens is 80% of the people are being considered to be as secretors, and it is inherited by the presence of an allele called SESE or SESE. So if you are having a capital SE allele, okay, then you will be, that is, all your body fluids will be containing the ABO antigens present in it. Okay, and if you are having another phenotype called the small letter SESE, you will not have the Now, if you're having a small letter SE gene, SESC, no antigens will be expressed in the saliva or the body fluid. Now, why do I have to mention this over here? It is also giving you another mechanism. That is, if you get a body fluid, you can also use it for, if the person is a secretory positive person, you could also find out what is the possible, what blood group of that particular person because of the presence of the antigens in their soluble, which have been present in the secretory fluids. So with this, I guess this is clear to you. And we could also talk about two more things called the pseudoalleles as well as the isoalleles. Now, as I mentioned to you earlier, in the case of multiple alleles, we do express that the allele should be present in the same locus of the homologous chromosome and no crossing over should take place between them. But there are certain regions where what happens, the alleles might show recombination with each other, and so the term of multiple allele cannot be used for them. In such cases, we call them as pseudoallelism. So it's a state in which two genes with the similar functions are located so close to one another on the chromosome and they are being genetically linked. And they are nearly inherited together. But sometimes what happens, rarely, they would also recombine. So one hypothesis is that pseudoalleles are formed as a result of the gene duplication events, and duplicated genes can undergo gene evolution to develop new functions. Now, for example of the pseudoallele is in the case of Drosophila red eye color. They affect they have different mutants like white and apricot and all that, and they affect the pigmentation of the eye, and they have the same character, so they are allelic. But sometimes they could also undergo recombination, and hence they are considered to be as non-allelic. So, red eye color in Drosophila, some variants might serve as pseudo-alleles rather than considered to be as multiple alleles. Not all the forms, but sometimes if they recombine with each other, then they are considered to be as pseudo-alleles rather than multiple alleles. And the characteristics of pseudoalleles is that they are closely linked allele within which crossing over can occur, and they affect the same characters. In addition to this, we also have to mention about isoalleles, the alleles which produce the same phenotypic effect in the homozygote, but which nevertheless differ, are referred to as isoalleles. Now, in the case of eye color, eye color, in the white eye color in the case of Drosophila, you can see that they might be originated in different ways, but they are not necessarily alike. The genotype, when you go to refer to the genotype, and such alleles are referred to as isoalleles. And mutant genes that give slightly modified phenotypes are also called as mutant isoalleles. So with this, we come to an end of the multiple alleles. Hope so it is clear to you. Thank you for now.